from Poseida, as a disclaimer, as a starting point, so we are a publicly traded company. So please refer to our SEC filings along with our 10Q and 10K for any risks and uncertainties. So at Poseida, we see ourselves really on a mission to redefine cell and gene therapy for patients with cancer, rare disease, and beyond. We look at ourselves as a truly engineering company with proprietary in-house technology that we've developed that really allows us to go after cell therapy and gene therapy. When we look at the cell therapy side, our focus really is on allogeneic. We believe it's the future, we believe it's better for patients downstream, and even just the cost models alone would dictate and warrant it. In August of this year, we announced a partnership with Roche to help advance our heme malignancy portfolio. And then on the gene therapy front, we're very focused on in vivo liver-directed gene therapy. So we have multiple technologies that we're utilizing in the gene therapy space to go after some of the hardest to treat rare diseases and in some of the toughest to treat patient populations. In October of 2021, we announced a partnership with Takeda to help accelerate and focus on our pipeline. And then the third priority that we really focus on is platforms and partnerships. We often get asked the question, why not to focus on just one or two things? And really the technology is so broad and it can go in so many different areas that we need partners at the table to help us accelerate and advance it forward. So when we talk about being a genetic engineering company, we're really utilizing three different technologies as our core. So the first is gene insertion, the second is gene editing, and then gene delivery. From a gene insertion standpoint, we're using Super Piggyback, which gets me really excited on most days because Super Piggyback is a non-viral transposon-based system. And what we're able to do with Super Piggyback on the gene therapy side is deliver sustained and permanent integration into hepatocytes. The other beauty of Super Piggyback is the cargo capacity is theoretically limitless. So we're able to deliver large transgenes without having to reduce or cut or knock down. And a great example of that is our P Factor 8 101 program. So that's for hemophilia A using the completely non-viral system. We're able to deliver the full Factor 8 transgene with all the regulatory components and not having to cut or truncate down. Super Piggyback provides us flexibility, so it gives us the opportunity to go after a broad range of cells, and we see significant advantages with tox, tolerability, potency, and also downstream cost of goods. On the gene editing front, this one also gets me very excited. It's called Cas-CLOVER, and it's a proprietary in-house built system that gives us the flexibility to do knockouts at a pretty high efficiency rate and potentially knock in for gene therapy. The beautiful part about Cas-CLOVER for our cell therapy side is it gives us the ability to really edit resting T cells while maintaining the desirable stem cell memory characteristics that we want to see. Major advantages on this system, potentially lower cost, tolerability, and multiplexing, which I'll talk about on a separate slide. And then the third bucket is, how do you deliver, how do you move it, in vivo? And that's really the core of where we're focused in gene therapy as well, is our nanoparticle technology. So Poseida is using biodegradable nanoparticles, which give us a whole range of potential options for therapeutic advantages, but it really allows us to deliver long-term stable gene expression. We'll talk about that in a moment. We're utilizing our in-house and partnered nanoparticles through both our non-viral system, but also what we call our hybrid system, which is using an AAV plus lipid nanoparticle. The beautiful part here, again, though, is the ability to deliver multi-cell types in target-specific tissues. So to spend a bit more time on Piggyback, so Piggyback on the cell therapy side, preferentially favors stem cell memory. And we believe stem cell memory is key and core to our allogeneic portfolio. And it works exceptionally well in resting T cells, providing for improved tolerability and a more durable response. Again, the great thing of Super Piggyback is the cargo capacity. So we're talking about single cars now with most therapeutic options. Think of multi-cars involving 2 or 3 potential cars built in. A lot of flexibility the system gives us as we continue to build our cell therapy portfolio. On the gene therapy side, what we really see the advantages of the super piggyback or piggyback system is that integration, moving the DNA and shuttling it in to provide stable and long-term disease correction. So we're talking about being able to rescue the phenotype of the disease and permanently integrate where you're continuously being able to express the protein required. Piggyback is exceptionally great, specifically in dividing tissues like those in juvenile and neonate, where most of the traditional transient AAVs just can't touch. It's a highly efficient system that potentially allows us the opportunity to provide a single treatment functional cure or redosing to get to the titration level of a specific PK level for a patient. Again, large cargo capacity here as well for gene therapy, and we've shown delivery both with our nanoparticle program, so that's the P-Factor 8 101 again, but also with our POTC 101 program utilizing the hybrid approach. Cas-CLOVER. So Cas-CLOVER is a proprietary technology, as I mentioned, that was built in-house. We believe it's the cleanest gene editing system out there. We published a paper earlier this year on Cas-CLOVER, so I would encourage you to have a look at it because it does speak about the high fidelity nature of our system. Cas-CLOVER, the beautiful piece of Cas-CLOVER is we're using a deactivated Cas9 coupled with the guides, and at the center fused with this beautiful enzyme called CLO51, which really allows us to do most of the editing. It's important because this allows us to edit resting cells for our allogeneic program, and we're looking at utilization of the Cas-CLOVER system with gene therapy. So think knockout targets that you need to get an 80 or 90% knockout on to deliver therapeutic levels, or a knock-in potential with the system. What we've seen with Cas-CLOVER as published is low to almost no off-target cutting, high editing efficiency, and multiplexing capabilities. All right, so we talk about disruption, innovation, and CAR-T, and what we really mean by that is allogeneic CAR-T for oncology. And there's really three things that we think about when we talk about disruption. So the first is cell type matters, and we really believe that stem cell memory has the potential to move the allogeneic program forward. It is the ideal cell type for CAR-T due to not only the greater safety profile, but also for durability. Self-renewing, long-lived, and multipotent. We believe with our allogeneic program, we're also able to address graft versus host and host versus graft alloreactivity with our gene editing technology. And our booster molecule technology potentially has the opportunity to deliver hundreds of doses from a single run, translating into significant cost savings downstream, but also broader patient population reach and huge attractiveness from a commercial standpoint. Disruption in gene therapy for us is about achieving fully integrated technology. So what we mean by fully integrated, again, is rescuing the phenotype, but doing it in a way that course corrects the disease permanently. Piggyback integrates into the DNA to potentially allow for single treatment cures. We are utilizing piggyback and nanoparticle, again, in our hybrid system, which we believe can actually help with many of the issues that have been seen with the traditional AAVs and broad application given the cargo size. So as we move towards a world of non-viral, which is our stated goal as a company, we originally started by looking at a dual AAV system with viral, so combining the DNAs on both sides in our super piggyback and piggyback system, so transposon and transposase. That had high affinity and it looked great, but we felt the need to shift to the hybrid system where we can really take advantage of trying to drop the titers to magnitudes lower of all the other companies. So with our hybrid system, we're using a nanoparticle and an AAV, and we have data published so far. We showed it at R&D Day earlier this year on our OTC program that is allowing us to drop the titers by magnitudes lower than most traditional AAVs can offer. And we're moving forward pretty swiftly on our non-viral system. The first publicly disclosed program on that is the Factor 8 gene therapy for Heme, and that's in partnership with Takeda. All right, so I'll talk a little bit about the power of partnership back to, the initial slide here. So in August of 2022, we announced a collaboration with Roche to really help advance our heme malignancy portfolio. We're very fortunate to have Roche as a partner at the table with us. And then in October of 2021, a partnership was executed with Takeda to focus on liver-directed and HSC disorders with 6 targets and an option for 2 more. I'll touch on briefly the Roche partnership as it has recently come out, and we've received quite a bit of questions on it. So I'll take you to the right-hand side with Roche. So it gives them a license for BCMA allo and CD19, CD20 allo, which we're very excited about both of those programs, along with options to 2 other heme programs. From a biobuck standpoint, it's a $6 billion in economics with an upfront of $110 million and $110 million in near-term payments. On the Poseida side, the beautiful part of the deal is it continues to give us access to our solid tumor portfolio, and we announced earlier this year our first allogeneic solid tumor program, MUC1C or MUC1C, that was initiated this year, and we're very excited to see that program move forward for patients. And we retain rights to all of our solid programs, which really preserves what we believe a lot of potential commercial upside. This is just a look at our pipeline. So we are allo pretty much all day, every day. We have 1 program still in auto, so that is the PSMA 101 program. And if you can say that 10 times fast, you get a gold star for the day. But very exciting portfolio here. We look forward to seeing it continue to advance both on the Poseida side and with our partner Roche. All right, so I'll touch briefly on the Takeda partnership. It was announced in October of 2021, but we feel most people might have missed the importance of this partnership. So it's a strategic partnership on our in vivo gene therapy, 6 initial disease targets, including our hemophilia A program, which is, again, utilizing our non-viral technology with an option to add 2 additional targets. From a biobuck standpoint, it's $3.6 billion in potential milestones plus royalties. From the Poseida side, on the left-hand side of the slide, it gives us the opportunity to continue advancing our POTC 101 program, which is utilizing our hybrid plug-and-play system, and access to all other liver and HSC-directed targets. And if we know liver-directed targets, there are several hundred that have potential options. We retain rights to our all other tissue targets, which we are actively looking at what's beyond liver, and rights to our ex vivo gene therapy applications, should we choose to pursue it. So a smaller pipeline slide compared to what you just saw on the cell side, but we're just getting going with gene therapy. Our 2 lead programs are POTC 101 and P-factor 8 101, and we are actively looking at what's next in liver-directed. All right, so we're just beginning to tap the potential. Back to my initial comment, why not just focus on a few things? Our technology is so broad and expansive that there are multiple areas of untapped potential, not only for patients, but for the systems overall. Again, we are very fortunate and happy to have Roche, a leader in oncology, and Takeda, a leader in rare disease, sitting at the table with us. I think I did that shorter than I had anticipated. So thank you for the time and attention.